Hello, everybody. Today, I would like to share with you all with the Term 1 Biology, Stpm Biology, Term 1, Chapter 1 to 3. I'm Mr. Ong from College Tingkatan 6 Seri Putra, Ipoh, Perak. Good afternoon. So, next week is the Stpm Biology Term 1 examinations. So, today I would like to share with you learning biology through question for Chapter 1, 2, and 3. Let me start first. Okay, Biology Term 1, Chapter 1, 2, and 3. Chapter 1 is biological molecules. Chapter 2 talk about structure of cells and organelles. Whereas for the chapter 3 is membrane structure and transport. Now, in chapter 1, biological molecules, there are many subtopics in this chapter. So today, I just focus on several topics, which is what we call quite important, and then the student may not understand very well. So for example, describe the structure of water molecule, relate the properties of water to its biological significance, describe the formation of sucrose and explain why sucrose is a non-reducing sugar, and explain the basic principles of electrophoresis for protein and nucleic acid separation. Once you know how to describe the structure of water molecule and relate the properties of water to its biological significance, students also are able to describe the structure for the rest of the biological molecules. For example, glucose, sucrose, polysaccharide, protein, lipid, and nucleic acid with the same manner. So the important thing is the student, you must able to learn and know how to answer the questions. Let us see the structure of water molecules. This is the first subtopic in the chapter 1. Normally, the student, they will just ignore it because it is the first topic, the first subtopic that study. And it is quite simple for the student. If the student, they say, this is just a water molecule. Okay, let me see how to answer or to write the structure of water molecule in Stpm level. Like as you know, water molecule consists of two hydrogen atoms combined with an oxygen atom by covalent bonds. So you must write the number, how many hydrogen atoms and how many oxygen atoms. And then they are joined or combined by covalent bonds. Then student need to write the water molecule is dipole due to uneven charge distribution, or we can say unequal sharing of electrons between hydrogen and oxygen atoms, as you can see here, okay, the sharing of electrons. And then oxygen is more electronegative than hydrogen. This is what you need to write. The hydrogen atoms bears a partial positive charge. So I highlight the partial, or the student, you need to write slightly positive charge. And then the oxygen atom bears a partial negative charge. So the words partials is very important. Then the next one is talk about the bond angle. of the water molecule form is 104.5 degree due to the repulsion of the lone pairs electrons. So there are two lone pairs of electrons here. So the repulsive force they form, so they are pull, the angles becomes smaller. So becomes a 104.5 degree. So this is the structure of the water molecules. Then the student need to relate the properties of water to each biological significance. So the terms of biological significance, they can be inside one cell or in a multicellular organisms or for the environment. So for example, water is a good or can use excellent or versatile solvent. Then we cannot use universal solvent because the water first thing they cannot dissolve the container. So this one is it is not a universal. So we should use water is a good, excellent, or versatile solvent, either one, due to its molecular polarity as a dipole. Then the oxygen regions of the water molecules which are partial negatively charged. Again, we use partial negatively charged. Clean or attract, either one, to slight positive charge polar molecules. So this is for polar molecule is also the slight positive here. Okay, so the partial negative charge, they attract to the slight positive charge. Whereas the hydrogen regions of the water molecules are partial positively charged, attract to the slight negative charge of polar molecules. So here can see this is water molecule and this is the ammonium ions. So you can see their polar charge. Okay. All right. Then for the ions or the salt, a hydration shell is formed. Okay, hydration shell is formed when water molecules surround the ions. For example, you can see here, this is they show how the salt is dissociated. Okay, dissociated, and then this is the cations. So they form the hydration shell. Okay, where this is the partial negative charge, and then here is the partial positive charge. And then due to the water solvent property, water can act as a transport medium because the polar molecules and the charged ion that dissolve in the water, and then the water act as a transport medium to transport the polar and charged particles or ions in organisms. Okay, for example, in the blood or in xylem and phloem. Then the next properties of waters is each water molecule can form maximum 4 hydrogen bonds as shown in this diagram. Okay. Then an oxygen atom in a water molecule has 2 regions of partial negative charge. As you can see, here they got 2. Okay. So this is the lone pair of electrons. And each of the 2 hydrogen atoms has a partial positive charge. Okay, the partial positive charge. So they can form attraction force. So this attraction force is called hydrogen bonds. Remember, the hydrogen bonds, we use the dotted lines to represent it. Okay, and the attraction force formed is known as cohesive force because the attraction force is formed between water molecule and water molecules. The water molecules also display adhesion, or we call the adhesive force, in which the water molecules form hydrogen bonds with charged groups of atoms or the molecules on their surfaces. So the combination of cohesion and adhesions accounts for capillary actions, and then to transport water and dissolve minerals. Okay, not only water and dissolve minerals as well against the gravities in plants. And then because of the transpiration, and then with the transpirational pull, so we'll pull the water and dissolve minerals up to the shoot. So due to the cohesions, the water has a greater surface tension. Okay, we must use the greater surface tension. So this will make the water as a coated with an invisible film. Some animals, such as insects, can stand, walk, slide, or run on water without breaking the surface. For example, the water strider here. Okay. Then the next properties, which is quite important, which is the water has a high specific heat capacity. So what is specific heat capacity? The amount of heat energy that requires to be absorbed to change the temperature of 1 gram of water by 1 degree Celsius. Okay, you can use a kilogram or gram. So here is joule or kilojoule. So this is the definitions. Whereas water has a high specific heat capacity, that means a large amount of heat, okay, is required to be absorbed to change 1 gram of water by 1 degree Celsius. So the amount of heat is 4.2 joule. Then why the water has a large has a high specific heat capacity? This is because much of the energy is used to break the hydrogen bonds formed between the water molecules. Therefore, as a result, the water as a temperature buffer is relatively slow to heat up or cool down. Okay, so what they show you here, okay, once from the 1 degree Celsius, okay, between 0 to 100. So if they want to increase 1 degree Celsius by 1 degree Celsius, so they require 4.2 joule. So this is the significant is the temperature buffer. So the biological significance, cells that bathe in water or aqueous environment have a stable surrounding temperature. And then the stable surrounding temperatures is important because the enzymatic reactions that take place in cells, they are sensitive to temperature change. In terms of organisms, water helps multicellular organisms to maintain their body temperature. So this one is for homeostasis. Then the ocean and other large water bodies have relatively constant environmental temperatures and suitable as habitat for aquatic organisms. Because the temperature of the ocean and other large water bodies, for example, lake, pond, they have the constant environmental temperature. The next properties is water has a high latent heat of vaporization. The definition of vaporization is the amount of heat energy that requires to evaporate 1 gram of water, okay, or it is lost when 1 gram of water is evaporated. So it's 2.26 kilojoule for 1 gram. So this is much higher than 4.2 joule. You can compare, look at here, this graph here. So in order to vaporize the water, okay, from liquid water to gases, large amount is required here. Okay, so the 540 calories equivalents to 2.26 kilojoule. So the explanation similar to the specific heat capacity. So they requires to break the hydrogen bonds between the water molecules and evaporate away into water vapor. So the significance in organisms, so there are 3 here, I listed 3. First, vaporization of sweat provide cooling effect for human to maintain body temperature. Second is in plants, the transpiration, okay, they also provide cooling effect to plant. And then the last one is for the environmental habitats, okay, vaporization of water from the large water body. So they will lost or carry away large amount of heat. So here can maintain a relative constant environmental temperature. So this one ensure the survival of aquatic organisms. The other properties of water is cold water is denser than ice. So this is occur in the this one is important for the organism that live in habitat or in the country which have 4 seasons. So water expands as it freezes because the hydrogen bonds joining the water molecule in the crystallized lattice keep the molecules far enough apart. So therefore, the volume of the ice is increases about 10%. So the density will be around 10% less than the density of water. So from this diagram, we can see when the water becomes ice, so there's forms like the crystalline lattice, so they have more spaces compared to the water in liquid form. Therefore, the ice floats in water. Why this one is so important? Because the floating ice insulates the water below it, preventing it from freezing. This one is for the large water body. So that this can allow the aquatic organisms to survive in the water during winter. So here they have during winter importance. Okay, then the other one is also related to ice, and then water has the highest, okay, the highest density at 4 degrees Celsius. So as the ice form on the surface of the water body, then water below the ice, the temperature will drop. Okay, they will drop. So when they drop to 4 degrees Celsius, the water at 4 degrees Celsius has the highest density, so will sink. And then the water with less dense than the 4 degrees Celsius water will rise up. So this will form a circulation. This circulation is important to enable the nutrients they can from bottom to water near surface. Then this also ensure the survival of aquatic organisms at different water level so that they can get the sufficient nutrients. The other one is water also as an essential metabolite due to dissociation of small percentage of water molecules into hydrogen ions and hydroxide ions. So the hydrogen ions and hydroxide ions are very reactive. So they participate in biochemical reaction of metabolisms. For example, they are used in hydrolysis reactions and also as a source of electron and hydrogen in photosynthesis. Okay. After the water properties and its biological significance, now we are moving to the next part, which is talk about sucrose, the formation of sucrose, and also why the sucrose is a non-reducing sugar. So in answering the formation of sucrose, student can write sucrose is a disaccharide formed from one alpha glucose. Remember, you must have alpha, okay, alpha glucose molecule and one fructose molecule by condensation. Okay, so in one sentence here, you must have alpha glucose and by condensations. If without alpha glucose, you cannot earn any marks here. Then, even though we already mentioned condensation, students also need to write a water molecule is produced. So remember, it's a water molecule, okay, a molecule. Huh? And you need to describe how the, what we call the alpha-1,2 glycosidic bond is formed. Okay, since we are using the alpha glucose, so it's an alpha-1,2 glycosidic bond is formed. Okay, So this is alpha glucose. So here is carbon 1. This is carbon 2. Okay, carbon 2 of the fructose. So the hydroxyl group, see, hydroxyl group of carbon 1 in the alpha glucose, okay, join or react with the carbon 2, the hydroxyl of the carbon 2 in the fructose, okay, to form alpha 1,2 glycosidic bond, okay, alpha 1,2. Maybe student prefers to use this diagram, okay, but students must understand that when they are using this diagram is where the sucrose, the fructose molecules is mirrorly arranged. So the carbon 2 is at this side. Okay, don't confuse. Here it becomes the carbon 5. Okay, for the fructose. No, so that's why I prefer to show this diagram, okay, rather than this. All right, then the sucrase, which is the enzyme, is required for the formation of sucrose. Okay, student always forget to write sucrase or the enzymes involved. And then you must remember this enzyme, they can catalyze the reversibility of the reactions. And this reaction condensation and the hydrolysis, this process is reversible. The next one, why sucrose is a non-reducing sugar? Because sucrose has no free ketone or aldehyde group. So the free aldehyde group in alpha glucose is at the carbon 1. And free ketones group is in the carbon 2 of the fructose. So since the free aldehyde group, yeah, and the free ketone group in the fructose, they are joined to form the alpha 1,2 glycoside bond. So there are no more free ketone or aldehyde group. Therefore, the sucrose unable to reduce copper 2 ions in the Benedict solution to copper 1 ion. In contrast, the maltose, even though here they form alpha 1,4 glycoside bond, but there is one free aldehyde group in the carbon 1. So maltose is a reducing sugar. So, and here, you see, I highlight the reduce, unable to reduce the copper. So it is the correct word is reduce. Don't use change or convert. Okay. If you know the reactions, the type of reaction, either reduce or oxidize, please use oxidize, reduce and oxidize, or decarboxylated or phosphorylated. Don't use change or convert. Electrophoresis. Explain the basic principle of electrophoresis for protein separation. So now we talk about protein separations by using electrophoresis. So the correct answer is polyacrylamide gel electrophoresis, or PAGE, PAGE. It's used to separate protein by mass or by mass to charge ratio. Because the polyacrylamide gel electrophoresis, they have smaller pore size compared to agarose gel. So the PAGE or the polyacrylamide gel electrophoresis is suitable to be used to separate protein. So first, see the protein here. Then the proteins react with anionic detergent or sodium doxycyl sulfate (SDS) to form negatively charged protein here. See, this is the SDS molecule. So they react with protein or polypeptide, so they will form negatively charged protein C. Okay, so this is a larger protein size. This one is smaller. So that means the larger the protein size, the more the negative charge of the protein. Okay, so the larger the protein size, the more amount of SDS bounds to the protein. Therefore, the negative charge on the larger protein is more compared to small protein size. Then after that, we load the protein sample, okay, in the polyacrylamide gel. Then in the presence of electric field. So since the protein is already negatively charged, so student remember, you must write in the presence of an electric field, the negatively charged protein, so they move toward anode, okay, anode. Or you can write positive terminal, okay, so that the negatively charged protein can move toward the positive terminal. Once the process done, the proteins with low molecular mass, they'll move further. Okay, they'll move further than proteins with the high molecular mass. So from here, by using the polyacrylamide gel electrophoresis, we are able to separate the protein. In contrast, when we compare to the electrophoresis that used to separate DNA, so we also using the gel electrophoresis, but you must mention agarose gel. Okay, we are using agarose gel electrophoresis, okay, to separate DNA fragments according to their size. First, the DNA samples are cut with restriction enzyme, okay, into many fragments with different sizes. Okay, so this is the DNA fragments that cut by the restriction enzyme. Then the sample, okay, these DNA fragments, they are loaded into the well at one end of the agarose gel. So here they have a lot of well, and an electric current is applied, okay, to move the DNA along the agarose gel. So since the DNA, student must mention DNA fragments are negatively charged. Okay, negatively charged due to the presence of the phosphate group. Okay, phosphate group is the negative charge. So the positive terminal or anode must place at the another end or the opposite end of the agarose gel, so that the DNA fragments can move toward the anode. So because all DNA fragments have the same amount of charge per mass, so smaller DNA fragments, okay, will move further compared to larger DNA fragments. Then the DNA fragments are stained with fluorescent DNA stains as they move through the gel, the agarose gel. Then after that, we expose the gel under the UV light. We can see the DNA fragments. So the DNA fragment was shown as DNA bands. So the further the DNA bands, that means the smaller the size of the DNA fragments. Or in other way, we can use DNA binding dye. This one can be used to, this dye can be used to stain the DNA and visualize it without using UV light. So that is for chapter 1. And in chapter 2, the structure of cells and organelles. So here I try to combine, okay, the different type of organelles functions into one. Okay, so in this part, today we will discuss about identify and describe the cellular components of eukaryotic cells as well as their function, and explain cell fractionation. So you can see this diagram. Look at this diagram. They show the sequence of events in the production and secretion of hormones by a cell. So here, student must understand the hormones here is talk about the polypeptide. Okay, the peptide hormones are peptide hormones. And then the process is start from the nucleus and then to the end. If the question says, takes 2 reasons why the cell is considered to be eukaryotic cell. So from the diagrams, the presence of nucleus in the cell and the presence of structure X or the Golgi apparatus in the cell. Then student also can write the presence of linear chromosome in nucleus. So don't write more than 2 reasons. The next questions, name the processes F and G. So the process F and the process G. Process F is transcription. If you don't know what is transcription, you can write synthesis of mRNA. And for G is translation or synthesis of polypeptide. Next questions, describe how the product of process G is transported to structure X. From G, okay, the product of G to structure X. Then you can write the polypeptide synthesized from process G is packed into transport vesicle. You must mention transport vesicle. Cannot say cannot just write vesicle. So which to be transported to structure X. You can say to the C phase of structure X also can. Name two processes to be occur in structure X. We know in structure X, the polypeptide processing or modifications occur. So in Stpm, if they ask two processes to be occurs in structure X, so it's glycosylation and proteolysis. Or you can choose the sulfation or phosphorylations. Describe process H. Process H. So here is at the end, the final product is hormones. So we write the hormones produced is packed into secretory vesicle. Then the secretory vesicle move and fuse with the cell membrane. They move and fuse with the cell membrane and release the hormones out of the cell by azocytosis. So must complete the sentence. Okay. The processes F, G, and H, okay, F, G, and H require more energy to proceed. Explain how the cell and particular organelles are specialized for this function. So this question, they got two parts. Explain how the cell, one thing, they're specialized for this function, and how the particular organelles. So the answer is the presence of abundant mitochondria, okay, in the cell. Okay, so we answer this part. And then the second part is mitochondria. The inner membrane of the mitochondria are infolded. Okay, infolded to form cristae. So you need to describe or explain why the cristae is important. This increases surface area. Okay, surface area to embed many ATP synthase and enzyme complexes. For what? For aerobic respiration. Or you can write for oxidative phosphorylation. Then more ATP molecules can be synthesized or generated more, and more energy can be provided for the processes. What are the processes? F, G, and H. Okay, so this is the answer. You cannot say the presence of mitochondria. You must have abundant. So to increase the surface area, okay, and more ATP, more energy. Okay, the next question, lysosome is produced from structure X. Okay, from the structure X here. Describe one function of lysosome in the hormone secreting cells. So lysosome fuses with autophagosome, containing worn out organelle or damaged organelles to form secondary lysosome. Dehydrolytic enzymes, must mention dehydrolytic enzymes within lysosome, hydrolyze, okay, don't say convert or change. Hydrolyze macromolecules of the organelles into their monomers. So these macromolecules are referring to the carbohydrate, okay, the polysaccharide, polypeptide, okay, fat, so such as glucose and amino acid for the monomers to be absorbed and neutralized by cell. Okay, or can be to be absorbed and reused by cell. So for this question, because we are focusing to the hormone secreting cell, so we accept autolysis function. But phagocytosis and apoptosis function are rejected here because this is referring to the hormone secreting cell, not phagocyte. All right. The other one is the cell fractionation. We want to talk about the cell fractionation. I think students also know what is cell fractionation, but actually the process, how to carry out the cell fractionation is not so familiar. So today we would like to discuss how the cell components, okay, and organelles in animal cells can be isolated or separated by cell fractionation. So the cell fractionation involves homogenization and centrifugation. So in homogenization, the animal tissue are homogenized by Pasteur homogenizer or by using blender. Okay, so this is the Pasteur homogenizer. So as genic as possible in a co-isotonic, so must mention co-isotonic sucrose solution. This one is to minimize the autolysis of cell organelles, okay, by the hydrolytic enzymes. Okay, then after that, what we get is the suspension. So the suspension form is filtered to obtain cell homogenate. Okay, then the cell homogenate is subjected to centrifugal force, or which is known as G force, by spinning in a device called centrifuge. Okay, a series of increasing centrifugal force is used to separate organelles of the animal cells on the basis of their different sizes, shapes, and density. Okay, remember, student, the centrifugation actually they separate the cellular components, okay, according to the sizes, shapes, and densities. So this is known as differential centrifugation. And in the differential centrifugation, centrifugal force separate the extract into two fractions, a pellet and supernatant. So the pellet is the bottom one, the zinc. Then the suspension here is called the supernatant. So after centrifuge, so this is called the supernatant 1. This is pellet 1. The pellet 1 will contain nuclei or the largest organelles. Then the supernatant 1, they contains other cellular components. So we transfer the supernatant 1, okay, to the new centrifuge tube, then increase the centrifugal force to spin it. Then they will form the supernatant 2 and the pellet 2. So the pellet 2 normally contain the next heaviest, larger organelles such as mitochondria, okay, lysosome, chloroplast. But since this question focuses on the animal cells, so we cannot write chloroplast here, right? Then after that, if you want to study the smaller cellular components, so you need to centrifuge again under higher centrifugal force, okay, to obtain the next pellet and so on. Okay. So the pellet that form at the bottom of the tube contain larger and heavier components such as nucleus, okay, nuclei. Then the supernatant, the liquid above the pellet, so they can contain the smaller organelles and the cellular components. So the supernatants can be centrifuged again, okay, at a higher centrifugal force to obtain a pellet which contains smaller cell components. So a series of pellets containing cell organelles of smaller and smaller size, shape, and density can be obtained and studied. So through this, a series of what we call the centrifugation, so we can obtain a different type of pellets. Okay. Then this centrifugation, okay, the settlement, or we call the sedimentation coefficients, the use is the verbal unit. Okay, the S unit. So remember the ribosome, we have 70S ribosome and 80S ribosome. So this is we are measuring the ribosome by using the verbal unit. All right. Now is the chapter 3, the last chapter for today's discussion. In this chapter, we will talk about the structure of plasma membrane and the importance of each component, okay, of the plasma membrane. And also, we will discuss how to calculate water potential. Okay, water potential of plant cells in solution of different concentrations. If the questions ask about describe the structure of plasma membrane with the aid of a label diagram, if they ask about diagram, so student, you need to draw the fluid mosaic model of plasma membrane. So the drawing is simple, it's like this. No need to draw the three-dimensional one. Okay, so in your diagram, okay, you must have phospholipid bilayer, this one, okay, phospholipid molecule, phospholipid bilayer. Then you must have integral protein, okay, integral protein, peripheral proteins, okay, outside the membrane, can be faces toward the aqueous environment or cytoplasm. Glycoprotein, glycolipid, and cholesterol. So you must be careful to the labeling. For glycoprotein and glycolipids, you must label the whole structure. You cannot just label points to here. This is oligosaccharide carbohydrate. Okay, so you must either point to or you highlight it. Cholesterol. The position of the cholesterol must in between two phospholipid molecules. Okay, and they got one part is close to the hydrophilic head, because here they got one hydroxyl group, which is a hydrophilic region of the cholesterol. For the description, if you want to describe the fluidity characteristic, so we need to talk about the presence of kink, or the presence of double bond of the in the fatty acid chain of the phospholipid molecules. So the function of this kink or the double bond in the fatty acid is to prevent close packing of phospholipids. So the phospholipid molecules freely to move laterally. This will give the dynamic, okay, of the phospholipid molecules. We talk about molecule. If you write phospholipid bilayer could move freely, this one is wrong lah. Actually, it's the phospholipid molecule. Okay, molecules. Then the phospholipid molecules can float from one layer to the other by enzyme, but this one is less frequent. But when we talk about dynamic or the fluidity of the plasma membranes, so it's more focused on here, freely to move laterally. So this is phospholipid molecule. Then if we say the lipid components in the plasma membrane, there are two. One is phospholipid, another one is cholesterol. So this is the kink, okay, or the presence of double bond in the fatty acid of the phospholipid molecules to prevent the close packing of the phospholipid molecules. So this is to increase the membrane fluidity. You can imagine that if the phospholipid molecule they arrange a lot here, so here they have more space, okay, due to the presence of double bond. So that the phospholipid molecules freely to move laterally, okay, freely laterally, okay. So the explanation, how to explain, so I present this the explanation for you so that you can understand more. Okay, can help you for the understanding. Phospholipid consists of hydrophilic head and hydrophobic tail. This one as we know. Okay, and then you must mention hydrophobic tail actually made out of two fatty acids. So then the fatty acids are such unsaturated fatty acid. Then they prevent the close packing of the phospholipid molecule due to the double bond. Okay, then the more unsaturated fatty acids to the hydrophobic tails are, so the more fluid of the membrane is. Okay. So in terms of the cholesterol here, okay, just now we talked about phospholipid, now we talk about the cholesterol. So cholesterol they found in between the phospholipid molecules. Then the hydroxyl group of the cholesterol associate with the hydrophilic heads of the phospholipid, as I told you just now. Then the hydrophobic regions of the cholesterol fit between the fatty acid hydrocarbon chains of the phospholipid molecules. See, they are found in between the phospholipid molecule, not in between the phospholipid bilayer. Okay, so this one you understand. So this is the cholesterol here. See, this is the OH, okay, which is I say the what we call hydrophilic regions. So if you want to explain how the cholesterol can stabilize the plasma membrane, so this is the answer. At moderate temperature, we cannot use high temperature or hot temperature and so on. Okay, because if you are using high temperature, the cell already died. Okay, then the plasma membrane already broken down. So what we can write is at moderate temperature, the cholesterol interacts strongly with hydrocarbon chain of the phospholipid because they are found in between here. Okay, so at moderate temperature, the phospholipid molecules should move loosely, they move apart, but because of the hydrophobic region, okay, hydrophobic region of the cholesterol, so they interact strongly with the hydrocarbon chain of the phospholipid molecules. Then they prevent the membrane from becoming too fluid. In contrast, at low temperature, the phospholipid molecules, they tend to pack closely. But because of the presence of the cholesterol, so the cholesterol acts as a spacer, okay, to restricting, okay, van der Waals interactions with the hydrocarbon chain of the phospholipid. So prevent the membrane from solidifying. So at low temperature, it increase the fluidity. Okay, cholesterol increase the fluidity of plasma membrane, whereas at moderate temperature, not high temperature, the cholesterol stabilize the plasma membrane by reducing the fluidity. Okay, once we finish discuss the fluidity, now we talk about the mosaic pattern. The mosaic pattern, we want to talk about the scattering arrangement, okay, of the proteins. Okay, integral protein and peripheral proteins. They scatter randomly, okay, in the plasma membrane. All right. So as I mentioned just now, say peripheral protein is attached outside the cell membrane is wrong because here is outside. Actually, is the both sides of the membrane. Okay, either is the aqueous environment or cytoplasm. Okay, are attached on the membrane while the integral proteins are embedded in the membrane. Okay, so the membrane proteins are amphipathic with hydrophilic and hydrophobic region. So the ones hydrophilic regions, they will contact with the aqueous environment and also cytoplasm. Whereas the hydrophobic region of the proteins, okay, the amino acids, they are embedded in the membrane. Okay, that means in the between the what we call in the phospholipid bilayer. Maybe the question will ask about the function or the role of the proteins in plasma membrane. So we talk about 5 major functions here. First, the protein can act as structural proteins by anchoring, anchoring the cell with extracellular matrix, or they can what we call anchor or connect with the other cells. Then another one is the transport proteins. So as a facilitator diffusion, in the passive transport and also active transport. This one you know lah. Enzymatic activity. Some of the proteins in the membrane that act as enzyme complexes, or enzyme, so they take part in a catalytic reaction. For example, in the mitochondria, you already know, in what we call the chloroplast, photosynthesis, and also there are many enzymes they found on the cell membrane. Then the next one as a receptor proteins. So receptor proteins, that means the receptor, for example, hormones or neurotransmitter binds to the receptor binding site. So they are what we call the signal or transmit the information into the cell, or they can have other cascade reactions occur inside the cells. Membrane protein, especially the glycoproteins, they will act as marker proteins, or we call the antigen for cell recognitions. So these are the function of the membrane proteins. All right, then we go to the next and the final part of today's discussion, which is talk about the water potential. So the calculation and the involved in water potential. So I retrieve the questions, from the past year, about the calculation part. An experiment is carried out to study the direction of water movement in plant cell. Slices of potatoes are immersed for 50 minutes in sucrose solution of different molarities at 25 degrees Celsius. So this one blah blah blah. The experimental data and the change between molarity sucrose solutions and the solute potential of sucrose solution is shown in the table below. So when you see here, the molarity of sucrose solution is 0.32, then the solute potential is negative 0.78. The percentage of change in mashed potato is 0. So that means this sucrose solution at this concentration is an isotonic solution to the cell set. So that's why there is no change in the mass of the potato. But when the mass of the potatoes increases, so that means here 0.2 molarity is the hypotonic solution. It's hypotonic to cell set. So that's why it's a positive 6.8. So the increase in the mass, so they show the turgidity, the turgid. The cell is turgid. So the other one is here negative. They mean the loss of the mass. So it's hypertonic to the cell set. So in terms of the condition of the cell, here is turgid. This one, even though in isotonic, it is flaccid. Isotonic is flaccid, and the hypertonic is also flaccid. Calculate the actual water potential of the potato cells in isotonic solution. So in isotonic solution, they ask you to calculate the water potential. Water potential. So first, you must write the formula. The water potential is equal to solute potential plus pressure potential. So then the solute potential here is, we can get it from the data, which is the negative 0.78. And then the pressure potential is 0. Pressure potential is 0 because there is no change in the mass, and then the cell is in a flaccid. So flaccid cell, actually the water potential, the pressure potential is 0. So that means here the water potential of the potato cells is negative 0.78. So this one easier. Then calculate the pressure potential of potato cells in 0.2 molarity sucrose solution at equilibrium. Okay, equilibrium. So that means here the potato cells here, then we place in the molarity of sucrose solution. Then they ask you to find the pressure potential. The calculation actually is quite easy. Okay, so in 0.2 molarity, now we look at the solution only. The water potential in the sucrose solution is equals to the solute potential and pressure potential. So from the data, we know the solute potential is negative 0.5, and then the pressure potential is 0 in any solution because there is no cell wall. Okay, so it's 0. So the water potential for the solution, okay, 0.2 molarity solution is negative 0.5. Okay, this one is important. And then the word here at equilibrium. At equilibrium, this one is very important because at equilibrium, the water potential of the cells is equal to the water potential of the solution because they are at equilibrium. Okay, so when at equilibrium, that means at the end, the water potential of the cells is equals to the water potential of the solution. Okay, then because now we want to calculate the pressure potential of the cells, so the water potential of the cell is equals to the solute potential plus pressure potential. So from here, because at equilibrium, here is equals to this, so the water potential of the cell is negative 0.5. Then the solute potential. The solute potential is we get from the previous calculation. So the pressure potential is blah, blah, blah. You get 0.28 megapascal. Positive value for the pressure potential. Okay, maybe you're still confused, but why we want to use the previous calculations? Okay, so don't worry, look at this slide. Okay, this is pure water. In pure water, everything is 0. Okay, so the water potential for pure water is 0. The highest is 0. Okay, in 0.2 molarity sucrose solution, so the solute potential, okay, from the data, from the table is negative 0.5. So the pressure potential is 0 because no cell wall here, okay, no any wall. Then the solution, the water potential for this solution is negative 0.5 plus 0. So what you get is here. Okay, then just now in isotonic solution, in isotonic solution, the water potential of the cell is equal to the solute potential. Okay, because the pressure potential is 0. Okay, so what we get is, see, the water potential for the cell is negative 0.78. Then this one is different to this because this one, the equilibrium is in isotonic solution. Now, when they reach equilibrium, the equilibrium is at 0.2 molarity. So therefore, the water potential for the cell is equal to the water potential for the solution at equilibrium. All right. So what is the cell? The solute potential of the cell? So it's at the isotonic. Okay. See here. Solute potential is equal to negative 0.78. So we take this one. Then we put into the formula. Then we calculate. So we get the answer 0.28. Okay. So this is how we get the answer. So remember, at equilibrium, the water potential solution is equals to the water potential of cell at the equilibrium, not this equilibrium, not this equilibrium, it's this equilibrium. So since you understand this concept, then we can move to the next part. If I add the question like this: 0.4, okay, which is hypertonic to the cell set. Then the solute potential is negative 0.97. So by using the same concept, so in this solution, the water potential solution for this one is negative 0.97. So at equilibrium, sure, at equilibrium, so this water potential of the cell must be same to this, equal to this. So negative 0.97. Okay, and then the solute potential is from the previous calculation again, negative 0.78. Then you calculate, what you get is negative 0.19 megapascal. Okay, in calculation, from calculation, but can the pressure potential value be negative 0.19 here, not in xylem vessel? Cannot. Okay, cannot. Why cannot? Okay, why cannot? So this one again a calculation. So this is because when the turgid cell is placed in hypertonic solution, the plant cells undergoes plasmolysis. Water diffuses out of the cell into hypertonic solution by osmosis. You see how I write the answer, okay? Full sentence, okay? What happened to the water? The water diffused out. From where? Okay, or to where? And then by what process? Then its contents shrink and the plasma membrane separate from the cell wall. So when they start to separate, the plasma membrane start to separate or pull away from the cell wall, actually the pressure potential already reach zero. So once the pressure potential reach zero, so the pressure potential cannot decrease anymore. Even though in calculation we can get negative 0.19, okay, but in reality, once they reach zero, and then we cannot get any calculation anymore or any figure. Okay, as the sucrose solution molecule increases from 0.2 to 0.4, because when they become zero, so it's zero lah for the pressure potential. Okay, but in calculation, they still show you like this in calculation. Okay, so I think today is I think enough for today's. So I hope this around 1 hour's sharing sections, okay, can help you or guide you in answering your Stpm question. And then the question actually is quite similar. So it's only the way how you answer the question. Okay. So remember, Stpm, the answer, they require a comprehensive answer. Okay, compact, simple, compact, and comprehensive. Okay. So, and then good luck for you all. And then any questions? Okay. The student asked differential centrifugation or self-fractionation. So self-fractionation, like if you go through my slide, you can see self-fractionation consists of homogenization and differential centrifugation. So actually, they still got others centrifugation method. For example, the sucrose gradient centrifugation. Okay, Subhashini asks how to score essay part. First of all, you need to understand the questions, get the requirement of the questions, and then you must have the content. Sure, you must understand the concept, you must get the concept right, and you no need to write very long. I think your teachers also will guide you, okay, guide the students. Okay, normally they say one sentence, one marks, but actually sometimes one sentence can be 2 marks. Okay, so depends how you organize the answer. Okay, and then sometimes one sentence they have no marks because they need to have another sentence, okay, to help them to have to complete, okay, the marks. Yes, good question. Yeah, for this question already answered. I think got one student. This student also answered, yeah, for my slide. Okay. Okay. So got other questions? So let's say how to score. Like just already answered to you lah, how to score the essay. First, you need to read the question. Okay, to get the question. For example, like here, you see the components in animal cells. So if the question already mentioned animal cells, so you must organize your answer into animal cells. Okay. Or the other question, for example, if they say describe let's say light dependent, okay, light dependent reactions that occur in C3 plant. Okay. So when we say light dependent, normally they no need to mention what type of plant. So you must focus on light dependence only. If they focus on one, let's say they ask you to describe the cyclic photophosphorylation. So you just focus on the cyclic photophosphorylation. No need to explain from A to Z. Okay. So this one for essay part, you must practice. Then refers to your teachers, see whether correct or not. Because sometimes you cannot focus on the first, you cannot tackle the question, and then maybe you don't have enough content. All right. So let's say if you got another questions, like you can contact me, okay, for the question. So I will try my best to help you. Okay. So for the chapter 4, 5, and 6, you can go through my YouTube channel. So I will try to post a video regarding the chapter 4, 5, and 6. For self-fractionation, they must going through again the homogenization. Sometimes they also call the centrifugation as ultra centrifugation because they are using they spin under the very high what we call centrifugal force or G force. Okay. For my YouTube channel, you can search my name, but be careful because there is another one. I don't know who have the same name and also at the end also end with MOE. So the one they have a lot of internet games. So don't subscribe the one. The one's a different person one. Okay. In my channel, I have several videos. And then they got here got one student already go through the video. Okay. Any questions? If no question, can we end the section now? So if no questions, so that's all for today. Thank you very much. And thank you for your listening. So anything you can go to find me. I think very easy to get me in YouTube. Okay. So that's all for today, and thank you very much.
Okay, Cikgu Ong. Thank you.
Ya, okay.
Atas
Sama-sama.
perkongsian tadi. Okay, terima kasih banyak-banyak. Jumpa lagi Cikgu Ong.
Ya, okay, terima kasih.
Okay, bye.
Bye.